Nuovo, io sono Anna Ambrosini di Fondazione Teleton ed è un grande piacere per me essere qui anche per
Foundation. And I'm very pleased to be here because, as Alberto Fontana was saying today, we have started a path with the GFB group for some time now. So it is again a pleasure to be here all together for the first time. My co-chair is Massimiliano Cerletti, whom I immediately introduce. He will be the first speaker of this session. Dr. Cerletti has a PhD and for several years now, unfortunately for us and for Italian research, he is very international and he has been living in the United States for some 10 years now. He's a group leader at Harvard University. And however, he was able to clone himself since he also has a very important activity at Progenitor Lab within GSK in the UK, and today we put the two souls of Massimo all together or with us today, and he will tell us about muscular stem cells and tissue reparation. Here you are. I'm just trying to understand how the pointer works. I wanted to thank the organizers. It is a pleasure for me to go back to Milan since I started from the Neurological Institute Pesta in Milan. I studied in Pavia and in Milan as well. So getting back to Italy is always a great pleasure for me. I actually work on stem cells, muscle stem cells. And this is the first slide, which is pretty important to understand what a stem cell is. What distinguishes a cell from a terminally differentiated cell? The properties are mainly two. The stem cell is able to self-renew, that is, to reproduce itself or to differentiate into mature cells. And this is a characteristic that actually distinguishes a stem cell from a mature cell. There are two types of stem cells: embryonic stem cells. You see here that the cell is practically obtained in vitro from the embryo, and again in vitro it is possible to differentiate it to make it into neuronal cells, blood cells, cardiac cells, and other tissues. And these are called embryonic cells, and their function is that of being pluripotent. So the question we are asking is this process reversible? From a stem cell which is usually gets mature and differentiates, is it possible to get back? This process is reversible. And Menacha in 2006, there was the identification of 4 factors that, when expressed in a differentiated adult cell, this cell reprograms and creates the so-called induced pluripotent stem cells, IPS. In 2002, together with Gordon, Yamaka won the Nobel Prize for Physiology and Medicine. This is an important process from the biological point of view because that allows the cell to go back to its embryonic-like state. So we can have a cell from the adult tissue, induce the factors, and this cell can actually take up some characteristics that are embryonic characteristics. Why is this useful? Because this can differentiate. So we can take the cell of a patient, induce it to its embryonic state, and thus move this cell to start differentiating again in all the patient's tissues. Well, I just wanted to hint at the fact that Plasticell is a biotech industry that developed this method to differentiate embryonic stem cells or embryonic-like stem cells into mature adult cells that exploiting recombinant chemistry. We were able to originate some precursor cells that express PAX7 and myosin. Why this? Because there are several applications. One, as you can see here, might be stem cell therapy. We can differentiate iPS or embryonic stem cells into differentiated cells, thus creating tissues, or there are the so-called adult stem cells, which I mostly work with. So adult stem cells, these are cells that are present in all the tissues of an adult, and they may be localized within the organ or outside the same organ. As regards skeletal muscles, stem cells, they are localized in a special region of the muscle fiber. They are also called satellite cells due to the fact that they are localized in a region between two fibers. One which is basal lamina and the other one is sarcolemma. And they have a special structure. They are encapsulated in these two membranes, and as you can see here in this electronic microscopy, they differentiate from the two fibrous nuclei. So, what's the process like? Normally, when there is a damage, a lesion on the muscle fiber, these cells are activated, they proliferate, and at that point, they decide their own destiny. Self-renewal, that is repopulating the niche with stem cells, or differentiating the neoblasts, thus repairing the muscle. Normally, this process actually takes place in two ways. Sorry, a glass of water, please. So the process takes place in two ways, through the fusion of a damaged fiber, or the same cells fuse together to create new fibers. This is a biological cycle that occurs during the damage of the muscle. So there are three groups that during the past few years have isolated satellite cells through FACS-CORT, through self-surveillance marker. So it is possible to purify and isolate from the muscle a specific population. There is this group in Stanford, it's the group of Ellen Blau, Amy Wager in Harvard, and a group with Margaret Buckingham in Paris. Also, the first question we actually asked, do these cells have the possibility to regenerate the muscle and repopulate the niche? We did these initial experiments isolating cells in green mice and injecting them in the red mice just to check whether they can actually perform as stem cells. As you can see, we can obtain some clusters with green fibers due to the donor stem cells that can actually regenerate the muscle in the host. We distinguish three types of fibers. Red fibers, there are those of the host. Only red fibers donated by the donor, and mixed fibers, green and red, due to the fusion. This is one of the first properties to define whether the stem cell actually performs as for regeneration. We used ambiax mice to check whether they can re-express dystrophin, which in these mice is almost lacking. Again, we isolated from GFB mice. We injected the cells in the dystrophic mice, and you can see that the green fibers can actually re-express the missing protein in the dystrophic mouse. Not only do they express dystrophin, but they can also act as in the self-renewal form. So we have a green cell, a donor cell, that re-expresses one of the typical markers of PAX7. So cells do not only differentiate and create a muscle fiber, but they can also repopulate the niche of stem cells. We also carried out other experiments with calorie restriction mice, and we are able to change their metabolism. Calorie restriction is actually a methodology that reduces in the diet of those animals the caloric intake. And when we transplant cells isolated from mice under calorie restriction and we transplant them into dystrophic animals, we have an increase in the amount of dystrophin on the fibers. So not only what I mostly deal with, that is trying to study stem cells in mice and now also in humans, satellite stem cells in humans, but we also want to pursue another approach, which is that related to drug therapy. With my collaboration in Progenitor Labs and Glaxo, we are trying to identify new drugs. So the fact that we are using techniques like combicult that actually allows us to grow our PS embryonic cells not on plastic but on beads. And through this combinatorial system, we are able to identify the pathways differentiating the cells, in my case, muscle cells. We are using these patient-derived cells to identify new chemicals. So the approach is that to differentiate cells, thus obtaining muscle progenitor cells, and then pass on to tribute screening, because we have interesting techniques that thanks to GSK we can investigate on biological processes like proliferation, the cellular cycle. The differentiation of these precursor cells are specific in the patients and thus identifying new molecules that may play a role. I, of course, wanted to acknowledge Amy and all the people in Harvard, in Glaxo, those from Progenitor, all the collaborators, and the facilities that were very useful during the past years for me and all my collaborators, and of course the agency families and Abby.
Grazie anche per essere stato nei...
Thank you very much for being here and respecting also our deadlines, the times that we have decided. Is there a question? Yes, there is a person that wants to ask a question. How many cells and how many patients do you need to generate iPS cells? This is a very good question because in mice we use a very limited number of cells, 100,000, and we want to prove that these are differentiated cells, so they have an increased functionality in comparison to a myoblast and so on. Of course, if we want to carry out experiments in humans, the situation is quite different, but as far as trials on animals are concerned, we want to use a very high numbers to differentiate the cells. Let's take, for example, the trials that we mentioned before. If we take the green mice into account, we cannot use the same strategy also for humans. So I think that I will use a greater number of cells as far as the human clinical trial are concerned in order to identify the cells. Yeah, it works. I just wanted to know if you spend a few words about the effects of calorie restriction. And the other question is, as far as iPS cells in humans are concerned, do you think that you will use its endogenous transplantations, or do you use cells from the patients? Well, of course, the idea of iPS cells and especially the widespread of these type of iPS cells in California is booming. But of course, they are important because they can be renovated into and renewed into tissue cells. As far as the application is concerned, we are of course late because there are no protocols that can be properly used. Of course, one of the most important protocols as far as the transplantation of iPS cell into mice is concerned is the teratome. This is the effect. If you don't have any teratome, you cannot obtain any particular efficacy. So every type of trial is carried out in mice, and when we have human cells, they are not properly studied, especially as far as iPS cells are concerned. Let's now take into account the calorie restriction. We are now studying the aging related to the calorie restriction.
Restriction prolong the lifespan and ameliorate the life and the quality of the animals. But so we want to study what happens in stem cell metabolically, and we see that there is an increase of mitochondria, functionality of the cells, number of satellite cells in the fiber, and many other different things. So.
Un'aggiunta a questa domanda.
I would like to add an element. Is the autophagy involved and related to the calorie restriction? Yes, I don't know many things about this aspect. There is a colleague of mine in the US who is studying about this and who is also studying in depth the idea of mitochondria and so on. Is there any other question? No. So thank you very much, and thank you for your presentation and also for your language.